Hello, everyone. I'm Peter Midford, and I will be the host for this webinar series. Our first presentation will be by Dr. Peter Karp.
Thanks for joining our BioPSYC webinar. All right, I'm Peter Karp from the SRI Bioinformatics Research Group. I lead the BioPSYC project and want to thank you for joining this BioPSYC introductory webinar this morning. Let me tell you a few brief words about SRI. We're a private nonprofit multidisciplinary research institute located in Menlo Park, California, about a mile from Stanford. SRI was started as part of Stanford many years ago. And we have two main partners in producing BioPSYC. One is the Paulson Lab at Macquarie University in Sydney, and the other is the Collado Vides Lab at the National University of Mexico. I'll note that two of SRI's past inventions include the computer mouse and the Siri personal assistant. Well, let me start off with an overview of the BioPSYC web portal and database collection. BioPSYC is a web portal that integrates both genome and metabolic pathway data with a really extensive array of bioinformatics tools that our users tell us are easy to use. It's an encyclopedic reference source that currently contains 19,000 total databases for organisms with sequenced genomes. 60 of those databases are curated with their contents derived from more than 130,000 publications. And you can get at a list of the curated databases here. I'd particularly encourage you to check out that list to see if your organism of interest has a curated database. And among the use cases for BioPSYC are basic information finding tasks, finding out information about the role of a given gene or a given metabolic pathway, finding out how a given gene is regulated in a given organism. Transcriptomics and metabolomics data analysis are other major use cases for BioPSYC, as is sequence analysis. And we have a number of comparative analysis tools. And many of our users work in synthetic biology. Now, all of these 19,000 databases, which we call pathway genome databases, combine a number of different types of data. They contain metabolic data about pathways, reactions, and metabolites. They contain proteome data about enzymes, transporters, and other proteins. They contain genome data, regulatory data, and in some cases, they'll contain gene essentiality data. And we divide these 19,000 databases into three different tiers. The very highly curated tier 1 databases, of which our E. coli database is one example, BioPSYC, and the tier 2 moderately curated databases, of which there are 60. And then the remainder of the databases are computationally derived with no manual curation. Now, a bit more information about these databases. We do have a number of eukaryotic databases for humans and for the major model organisms, of which some are curated. And we do have new databases for rat and worm planned to be released in the next month or so. But most of our databases are bacterial. And all of the databases listed on the right here are curated databases. And again, for the full list of databases, you can go to this link. And let me highlight one other database in particular, which is called Metacycle. Now, all the other databases in BioPSYC describe a single sequenced organism. Metacycle, by contrast, describes metabolism from all domains of life. It describes experimentally determined pathways, reactions, enzymes, and metabolites. It's a reference source on metabolism from all domains of life. It's a literature-based database. Metacycle does not contain computational predictions, and its contents have been derived from 68,000 publications. And one notable thing about Metacycle is it contains many different variants of a given pathway. For example, you may be surprised to know that there are 8 different variants of the TCA cycle that have been uncovered over the years, and they are listed in Metacycle. There are also 6 variants of L-lysine biosynthesis that you can find in Metacycle. So if you're interested in finding out about how a range of different organisms metabolize, say, a given compound, Metacycle is a great place to look. Now, just briefly, how do we create BioPSYC databases, and what is the range of data present in BioPSYC databases? Well, we start out with entries from GenBank, more specifically the RefSeq subset of GenBank. And we use the annotation present in GenBank, and then we add additional information to that database. We import data from a variety of different external databases. For example, we'll import regulatory data, subcellular location data, and protein features like enzyme active sites and metal binding sites. We'll also import gene essentiality data. We also run various computer programs that perform computational inferences, including prediction of metabolic reactions, transport reactions, prediction of metabolic pathways. We also predict operons. We have a new predictor for protein complexes that will be available shortly. And we compute orthologs among all proteins in BioPSYC, and we compute PFAM domains. So all of this is done automatically. Then for some of our databases, we'll perform additional manual curation to improve the databases further. Those are the tier 1 and tier 2 databases. Now, what do we mean by curation? What do curators do? Curators are PhD biologists who perform literature searches for new gene functions uncovered for that organism, new metabolic pathways uncovered. They author what we call mini reviews, short multi-paragraph reviews about the functions of a given gene or a given pathway. They'll update a variety of database fields like gene names and synonyms and activators and inhibitors of an enzyme. Curators will define protein complexes. They'll review and extend associations between enzymes, transporters, and the reactions they catalyze. They'll review the predicted metabolic pathways and review any false positive computational predictions. And for some of the databases, they'll enter regulatory information like transcription factor binding sites and promoters. So curation greatly improves the quality of data, the amount of data, and the accuracy of the data. Now, we actually have a subscription model in place for BioPSYC. Two of our databases are completely free, and they're a great place to explore what BioPSYC has to offer. And these are actually our 2 highest used databases, the EcoPSYC E. coli K12 database and the Metacycle pathway database that I just described. The subscription program supports our curation efforts for other databases, and there are actually 2 types of subscriptions. There are institutional subscriptions. You can ask your PI or your university library to sign up at no cost to you so that everyone in the institution can have access. And we have personal subscriptions available as well. But you also will receive a limited number of free page views to the subscription-only databases, again, to enable you to get to know the site and explore it. Now, one of the things we'll be covering in the tutorial is the rich range of bioinformatics tools available through BioPSYC. And these tools span genome informatics, pathway informatics, regulatory informatics. They also include microbiome informatics, comparative analysis, omics data analysis tools, and a number of advanced manipulation tools. And even in the 3-hour tutorial, we're not going to be able to cover everything here, but we'll cover a lot of this. And here's a brief overview of the tutorial series. Today's tutorial will cover basic encyclopedic search capabilities in terms of selecting a database, searching for information, what kind of information pages do we support, and then some sequence related operations like performing blast searches and extracting sequences from BioCyc. We'll also look at regulation and how you can be notified of updates in your area of interest in BioCyc. Then in 1 week, that tutorial will cover the Genome Browser and comparative operations and some data manipulation tools called smart tables, which users really love. They really let you perform operations that otherwise would take a programmer to perform, but you can do these interactively in a spreadsheet-like format. And finally, on November 17th, we'll cover a number of tools for exploring cellular networks in BioCyc, for analyzing transcriptomics and metabolomics data. And yeah, and you'll find additional recorded webinars online at this link if you want to explore additional topics that we haven't been able to cover in these 3 hours. Now, one of the take-home messages for today is to learn the main BioCyc information page types. And we'll be going through these in detail, but just as a preview, those main page types are the gene, protein, RNA pages, the transcription unit pages that depict regulatory information, metabolic pathway pages, reaction pages, and metabolite pages. And so if you weren't aware of one of these page types, you've kind of missed a whole dimension of information in BioCyc. Okay, let's stop there and go to the demonstration now. Little beach weather in San Francisco there. Okay, so biopsych.org is the homepage for our site. However, one of the really basic aspects of BioCyc is that almost always when you're searching against BioCyc, you're searching one of the 19,000 databases that it contains. And so generally the first thing you want to do is ensure that you've selected the right database as your current database. And you can see right here that my currently selected database is Helicobacter pylori. Well, let's say I want to change my database. Let me show you 3 different ways to do that. The first way is that we have a number of domain names defined that automatically select a database for you. So for example, if you're a B subtilis researcher, I can open bsubpsych.org, and that will go to a special landing page we've set up for B subtilis, and it will automatically select B subtilis as your current database. So this is a real fast way to connect to BioCyc. You're still talking to the same underlying site here. It's a real fast way to connect and make this your currently selected database. Another fast way to select a database is to go under the Sites menu and select one of these organisms listed here. These are all curated organisms. So for example, if you're a Mycobacterium researcher, you might want to connect to Mycobacterium tuberculosis, which again has its own domain name set up. Again, it set your current database to the H37RV genome. Now, if your organism of interest is not listed there in the Sites menu, another way you can change the current database is by clicking on this button, Change Current Database. And this window pops up here. And there are a number of ways that you can select within this window. You can simply type the name of your organism of interest. So if I want to find all the bacilli, I can type in bacillus, and we get a long listing of bacilli here. And I can simply click on one of these to select them. I can also click over here on the letters, this vertical row of all the letters, and I'll get all the organisms whose genus name begins with D here. Another fast way to select is to enter a strain name. And it will generally find the organism of interest really quickly that way by just typing in the strain name. And finally, you can select, there are some other selectors at the top that I'll encourage you to experiment with, including selecting by taxonomy and by properties of the organism. But a final way to select is to just click on one of the organisms in the left, and then you want to click OK. So now E. coli K12 is our default organism. Now, if you want to find out some basic information about the database you've selected, you can go under the Tools menu under Summary Statistics, and that will give you the organism summary page for this database. And there will generally be a summary text that's written here that says when this database was generated, what additional curation has occurred for it. There will also be a summary of properties of the organism, like is it an aerobe or an anaerobe? Is it a pathogen? Some basic statistics on its genome and its metabolic pathways are found down here. So this is the organism summary page. Now, the most frequent type of search that people do within BioPSYC is to look up a given gene to find out information about that gene. Oh, okay, then now I'm still connected to, well, I think it may actually be showing Mycobacterium by mistake. I think my current database may be E. coli. Let's select E. coli one more time to be sure. Okay. Right, one of the most basic search types that people do is to look for a gene of interest. So let's look for the E. coli gene DEOD, and I will hit return here. And we've now gone directly to the gene page for this organism, for this gene in E. coli K12. So again, the current database is always displayed at the top, and if you're searching a different database than you think you are, it's very easy to get confused about the results. But on this gene page, we get various information like the name of the gene and its product, accession numbers that's known by, locations of the gene product, and its map position on the genome, and the length of the gene and its product. We also get a list of reactions the gene product catalyzes and what pathways they're present in. And if we scroll down, we see a summary authored by, this is one of the mini reviews authored by our curators with many literature citations. So this gene has been the subject of many publications. And this diagram, called the regulation summary diagram, summarizes all the regulatory influences on this gene. We can see there are in green the positive acting transcription factors, in red inhibiting transcription factors, and CRP apparently is a dual regulator that both can activate and can inhibit depending on other factors. For other genes, we might have translation level regulators, although none are listed here. And we often have post-translational regulators. In this case, this gene product forms a complex. We also have various links to other databases. And notice here the different tabs that are available. We're looking at the summary tab currently, but all of these other tabs provide different facets of information. For example, the essentiality tab lists 5 different gene essentiality experiments, genome-wide gene essentiality experiments that have been performed for E. coli under different growth media, and lists when this gene was knocked out under a given growth medium, was there growth or not? And apparently, okay, went back by mistake here. Apparently this gene is not essential in any of these growth media. The reactions tab will list different reactions catalyzed by the gene product. This gene product catalyzes 7 different reactions in E. coli. And for each reaction, We're listing a range of information. What pathways is it present in? What's its KM? Is there experimental evidence for the activity of this protein, etc.? There's a protein features tab that lists various features on the protein sequence, including particular amino acids that bind, for example, phosphate. There are also PFAM regions listed. There can be enzyme active sites listed, cofactor binding sites, a range of things can be listed here. We also have an Operons tab that lists the operon that this gene is part of, as well as specific transcription units. So at the top, we see the overall operon, as well as, so DEOD is part of an operon involving 3 other genes, all listed in purple. And then there are other flanking genes outside the operon that are also listed. The diagram also lists transcription factor binding site locations, promoter locations, and here we've broken out the display for each transcription unit from each promoter to show in each case which transcription factors affect each promoter. And if I click on the promoters, I will go to another page type, which is the transcription unit page. So this is an information page that lists information about this operon, this transcription unit. And you'll see that there's a section in the page for each site in the transcription unit. For the promoter, for site number 1, which you can see is this DEOR site, has a little 1 after it. And so for each site, it tells us what transcription factor binds there. Does it repress or activate transcription? What is the sequence recognized by the transcription factor, etc., with some references at the end. And usually we will list evidence codes. So for this promoter, there's both experimental and computational evidence known. For these other sites, we don't have evidence. Oh, okay, there is evidence for the site listed over here. Again, both computational and experimental. And if we click there, we'll get more detailed information about the exact type of evidence. Okay, let's go back to the gene page now. So again, remember as you're looking at the gene pages to look for the information present under these different tabs. Now one of the other basic operations comes from this what we call the right sidebar menu. It's a menu of operations on the far right of the screen. And this menu of operations changes depending on what type of information pages you're on. So a metabolite page will have different operations here than the gene page does. And you'll see that we can retrieve the amino acid sequence of the gene product right here by clicking Get Protein Sequence. We can also retrieve the nucleotide sequence. Now this one's a little more involved. You can see by default, it's just added in it's shown us the nucleotide sequence for the coding region. But using this pop-up window, we can change the sequence region that's selected. So for example, if we want, let's say we want 100 bases upstream and 200 bases downstream, we can simply make those selections here. And then if we click, it updates the positions. And if I click Show Sequence, we now get a longer region with the start codon and stop codon shown in green and red. So that's a way to select an arbitrary region of the genome. Okay. Now, so far we've seen the quick search, which is a very fast way to do searches, but not a very precise way to search. Sometimes there are other types of searches that the quick search doesn't support. So if we look under the tools search menu, we'll see quite a few different types of search tools, and I'll show you a few of these today. if I click search on genes, proteins, or RNAs, we see a form that lets me do a lot of different things. We can essentially combine searches against different internal database fields of the gene or the gene product. Now, the simplest type of search is to search by a gene name or database identifier, protein name or database identifier. But we can search by other facets as well. So if I click here, it will expand this. And let's actually, instead of searching here, let's actually do a molecular weight search, where I'm going to search for all proteins with a molecular weight between 100 and 200 kilodaltons. And simultaneously, let's do a PI range. Let's enforce that the PI has to be less than 10 of the protein. As you see, we can also search based on, well, we'll come back to this. So if I submit that search, here's our result. We get a table of the resulting genes. And it shows both their molecular weight and their PI here. If we want to sort based on, say, ascending PI, we can click the little triangle there, and it will sort the table in that way. Let's now go back and let's close these and do a slightly different search. Let's say I want to search by protein feature. Let's say I want to search for all proteins that have a lipid binding site. I simply check this box here for lipid binding site and submit the search. And here's a list of 97 results, 97 genes that have in E. coli that have lipid binding sites annotated in the database. Now, another kind of search I can do is based on gene ontology term. Gene ontology is a way of annotating gene products according to their molecular function, according to which part of the cell they're found in, and according to which biological process they are involved in. And so I'm going to do a biological process search. I'm going to expand that hierarchy. This is a good way to find all the genes that are involved in some area of cell function. And in particular, I want to find adhesion. There we are. I want to find all the genes involved in biological adhesion. And I will submit that search. You can see there are other processes listed, such as carbon utilization, viral related processes, reproduction, locomotion. But let's do our search based on adhesion. And we get 63 genes involved in adhesion. And let's go back and do one more advanced search here. Let's close this one and let us search based on ligand. Let's see, here we go. So we can search or, ooh, okay, that's interesting. Okay. We can search based on, search for proteins where a given small molecule plays a specified role, either as an enzyme activator or inhibitor or cofactor, or a substrate, or as a transcription factor ligand. So let's enter arginine as a transcription factor ligand. And we get two results for the ARG-P and ARG-R regulators. Okay, let's now explore some other types of searches and some other page types. I'm going to do now a search for adenine. Now, previously when we did our search for DEOD, we went right to the gene page. But we're going to see now that adenine, searching for adenine gives us many results. And in a BioPSYC search, again, we're still searching within the E. coli database. But Our searches span many types of objects in the database. So we've searched against pathways and we've gotten pathway results. We've searched against proteins, we've gotten protein results. We've gotten results in metabolites, and we've gotten more types of results. Gene ontology term hits, and we've hit reactions that have adenine as a substrate, and we find growth media that have adenine in their name. We can click on any of these results to go to a result page, but let's focus for now simply on the metabolite adenine. I'm going to click here, and we now go to a metabolite page for adenine. And so it's showing us here the name of the object we're looking at, and it's verifying that we are in E. coli. This is the E. coli entry for adenine. There will be other adenine pages in different organisms in BioPSYC that show other information about adenine. Now, of course, the chemical structure of adenine will be the same in all the databases, as will the molecular weight and chemical formula and so forth, as will this other chemical information, including, for example, its SMILES and INCHI key, which are both ASCII notations for chemical structures. Like the gene page, the metabolite page has different tabs. Let's explore a few of those. There's the reactions tab. This tab lists all the reactions in E. coli that adenine is either a product or a reactant in. These are reactions that consume adenine. These are reactions that produce adenine. These are bidirectional reactions, both consume it and produce it, and these are transport reactions involving adenine. And okay, there's also a regulation page that shows enzyme catalyzed reactions that are known to be inhibited by adenine. So this one enzyme in E. coli is known to have adenine as a non-competitive inhibitor. Here are growth medium involving adenine. And this is just a larger, well, this is just another window to contain the structure. Sometimes the structure doesn't fit well up top. And this show all shows all those different tabs together. Makes it easier to search them in your web browser if you want to do that. By the way, for web browsers, we recommend Chrome and Firefox. Okay, let's search again for adenine. And this time we're going to click on one of the pathways, adenine and adenosine salvage 5. So that's the 5th variant of this pathway. When we refer to variants of a pathway, we say that 2 pathways are variants of each other if they accomplish the same biological function, but using a different set of reactions. Okay, so in this particular pathway, we can see adenine is being converted to INP. So this is a pathway page. This is another one of our major page types. If I use my mouse wheel, I can zoom that diagram a bit. Now on the pathway page, we can change the detail level of this pathway diagram. So if I want to add in structures for the main compounds along the backbone of the pathway, I do it with that selector. And if I click all compound structures, it will add structures for the side compounds as well, most of them. And you can see there's a summary authored by our curators about this pathway. This little diagram shows the locations on the E. coli genome of the genes involved in this pathway, followed by a listing of the operons involving the genes in this pathway. Okay, the pathway page does not contain any tabs. Now, from the pathway page, if I click on a metabolite, we'll go to the metabolite page for that compound. if I click on the reaction, we will go to a reaction page. So this particular, this page shows the catalysis of this reaction. It shows that there are 2 different isozymes in E. coli that catalyze that reaction. The compound is color-coded to show conserved moieties in this transformation. And we have various information like a standard change in Gibbs free energy from the reactants to the products. Okay, let's try going to a different pathway page here. And let's go to a more condensed display. So for this branching pathway, we're suppressing a good bit of detail. We're showing the main compounds only. We can add in the other compounds as well as the enzymes and gene names. Now, for some pathway pages, there will be an option to show regulatory information. I think for this pathway, no regulatory information is known. Okay. And let's go through the quick search a bit more. So if you leave your mouse over the quick search field, it will pop up this little window and tell you all the different things that you can enter in the quick search. You can enter a name or synonym for a metabolite, a gene, a protein, or a pathway. And you can also enter one or more substrings as well. So for example, if I want to find all the entities in BioPSYC that have that substring, we'll see that we get things like pyridoxal phosphate biosynthesis. We get a PYR operon leader peptide. We get a number of results. So you can search not just for single words, but for multiple words and for pieces of words as well, or substrings. You can also search for EC numbers. You can enter an INCHI key to search for chemical compounds. You can enter a BioPSYC internal ID for any metabolite or gene or protein or pathway. And you can enter identifiers from a number of other databases like Uniprot and KEGG and KEBI. You can search BioPSYC by their identifiers. You can also enter locus IDs for genes. Okay, and in Metacycle, let's quickly go to metacycle.org. So I'm again going to use the URL method of connecting directly to a database. So now my current database is Metacycle, and I should be able to enter an organism name here to search Metacycle. Yeah, so we've searched Metacycle for Mycobacterium, and we see quite a few different hits. And so we see Mycobacterium tuberculosis. We have quite a few different strains here. So I click on this one. Well, we would see pathways involved, but there are no pathways defined here. If we search for adenine in Metacycle, we will see quite a few more pathways than we found in EcoPSYC, because we're now searching against this universal pathway database, not just E. coli pathways, but pathways in all domains of life. And so if I click on adenine salvage 1, we get a pathway diagram. But we also, let's see, it also tells us some of the organisms that are known to contain this pathway. So this pathway is present in humans as well as in these 2 different bacteria. And well, actually, so I think this is an archaea. We can see that the expected taxonomic range of the pathway, namely the taxonomic groups we expect to find the pathway in, are listed right here. So this is some information you'll find in Metacycle, and but not in EcoPSYC. Okay, let's actually go back to E. coli now. And let's do a search for L-tryptophan. And again, we get pathway hits, protein hits, we get some RNA hits. So we'll see that there are pages in BioPSYC for RNAs as well. So here we're looking at the page for the tryptophanyl tRNA encoded by the TRPT gene. We see several known regulators that are known to affect this gene. But I also wanted to show you that if we go to the tryptophan biosynthesis page, again, we're on the pathway page. Okay, so here we can show the regulation details with this button. And we see at every step of the pathway, we see that the TRPR transcriptional regulator is known to influence transcription of this gene, as well as the tryptophanyl tRNA promotes premature termination of transcription via attenuation. So every gene in this pathway is regulated by both a transcription factor and an attenuator. Now, another thing we can do here is in the right sidebar menu, we can ask to show this pathway in what's called the cellular overview. So I'm going to click right there. So this is a diagram that depicts the full metabolic network of E. coli K12. And in this diagram, which is zoomable with my mouse wheel or with the little zoom bar to the left here, so the pathway we just came from, the tryptophan biosynthetic pathway, is shown here on the left. And in red. And in this diagram, each dot is a single metabolite, and each line is a single biochemical reaction. And when I mouse over a line or a metabolite, it identifies them in the pop-up window and tells me what pathway they're in as well. And if I zoom in further, eventually we're going to start to see metabolite names first, and then we will see gene names and protein names as well, enzyme names. And if I simply click and drag, we can move the diagram around. And the way the diagram is organized is that pathways are organized into larger groupings. So all the amino acid biosynthetic pathways are here. Nucleoside and nucleotide biosynthesis pathways are here. On the right side are catabolic pathways, such as carboxylate degradation pathways are here. And if we zoom in, we'll see which pathways we have here. These blue names are the names of the pathways. And also present in the diagram are periplasmic reactions are shown here. Shown here are cytoplasmic reactions that are not part of a pathway. And we're also showing transporters in the diagram. So for example, that is a colate transporter. And here we have a transporter for peptide antibiotics. Now, this diagram is generated automatically by our software tools from the underlying database. And we actually generate diagrams like this for every other organism in BioPSYC. So for example, Mycobacterium tuberculosis has a similar diagram. Since every organism has a different complement of metabolic pathways and reactions, we need to generate a different diagram for every different organism. Now, we we don't just have to use our mouse as the only way of searching the diagram. We can also use the right sidebar menu to search the diagram. So for example, if I want to find all the places that L-tryptophan is involved in E. coli metabolism, I can enter it as a search phrase, and let's make it a little smaller. Okay, so the search results are being shown in purple. So here's one purple tryptophan. Notice that tryptophan is showing as a triangle. Every triangle in the diagram is an amino acid. This is tryptophan, and it's in the tryptophan degradation pathway. So that's one place tryptophan occurs. And if we scroll back over here, we'll see another purple is the tryptophan produced by the biosynthetic pathway. Now, it's a little hard to see all the other purples in here. There are some. There's a tRNA right there. But if I use this little window here, lists all my search results. And so if I click on list, it will show here all the results that we obtained. And so if I click on the first one, it takes us there. If I click on the second one, okay, there's another tryptophan that's in a tryptophan synthase reaction. If I click on the third one, we go to a tryptophan transporter. A little hard to mouse over there, but that it shows us that's a tryptophan proton symporter. And we can do other searches as well. We can search for reactions according to the name or substring or EC number. We can search for pathways based on many different, for one thing, we could search by pathway. We can search for pathways by evidence. So for example, if I click here, okay, these are the different evidence codes that we can search for. Let's search for basically, we're going to search for inferred from experiments. So we're going to search for all the pathways that have experimental evidence. And we can see it's just about all of them because E. coli, and I would wager that the ones that are not shown in orange, probably somebody one of the curators forgot to add an evidence code, because the E. coli database is very, very highly curated. And so a very high number of its pathways will have experimental evidence codes. Now, to show you a contrasting diagram, let's go to Helicobacter pylori. And let's show its cellular overview. So if we go under tools, we can go directly to the cellular overview by going under metabolism and clicking on cellular overview. And you can see right away that Helicobacter pylori has much simpler metabolism than E. coli does, many fewer pathways. Again, we have biosynthetic pathways on the left, catabolic pathways on the right, energy pathways like TCA cycle down the middle. And okay. And we can search this diagram the same way as we did. We can search this, for example, we can search for L-tryptophan. And we're probably going to find fewer occurrences of L-tryptophan here than we did in E. coli. So there's one right there. In Helicobacter does have the tryptophan biosynthetic pathway. It does not seem to have a degradation pathway for tryptophan. It does have tRNA charging and this other reaction here. So more limited tryptophan-related metabolism. Let's see, I think I will give you a quick overview of the Genome Browser as well. So there are several ways to enter the Genome Browser. One way is if we go to a particular gene page, like if I type TRPA, we'll go directly to the TRPA gene page. and I can click View in Genome Browser right here on this button. And that will take me into the Genome Browser. And the Genome Browser has a couple different controls. At the top is an overview of the entire chromosome for this organism. I'm going to hide this menu, which is in the way. So the H. pylori chromosome is about 1.7 megabases. And the region we're looking at is this region in red right here. That's what's shown down below. And in this depiction below, the way to read this is like you read a book. We go across this first line and then back, and this same gene here continues across the second line, etc. So we're looking at a fairly large range of the genome. The genome we just came from, TRPA, the gene we just came from, TRPA, will be positioned in the middle of the diagram here and shown with the little hatch marks. And TRPA is part of the larger TRP operon. Well, all the genes in the same operon are shown in orange. So TRPA through TRPE, and then we have a different operon right here in purple. All these green genes are in the same operon. And as you can see, if I move my mouse over any gene, well, in this case, it's identifying the operon, not the gene. Okay, here it's identifying the gene for me. Okay, so there's a ribosomal protein in that gene, and it shows me the exact genome coordinates as well in that little tooltip. Now we can change the zoom level of the Genome Browser in a couple ways. We can directly enter starting and ending base pairs for the region we want to look at. That's one way. We can also, by the way, enter another gene name here if we want to search, reposition the Genome Browser around a different gene. We can also zoom in and out using this control here and move left and right. So if I click on the double arrow, we'll zoom in a lot. And if I click on the single arrow, we'll zoom in a little bit. And we can actually zoom all the way down to the sequence. So this control over here gives us a grosser control. So here we've zoomed in very far to where we see both the nucleotide sequence and the amino acid sequence is shown as well. And if we click up here, we'll zoom out a lot to what we call the operon level, so that we see a large set of genes simultaneously. And we can zoom it all the way out to show the entire genome in one screen here. Well, if we scroll down a little bit, we'll see it in one screen. In this diagram, again, colors represent operon membership. So for example, all these blue genes here are in the same operon. And direction of the gene represents transcription direction. And we do have a way of denoting RNA genes as well. They'll have a different arrowhead shape. I'm having trouble. Okay, there's an RNA gene right there. You can see how the arrowhead is at the top, not at the middle. So that is a tRNA coding gene. In fact, all the 4 genes in this operon are RNA coding genes. And that's an RNA coding gene as well. It's like a lysine tRNA. And yeah, in our next version, we'll actually have a way to search this diagram. That will be released soon. Now, in general, we tend to release new versions of BioPSYC 3 times per year, usually in March, August, and November, give or take. Sometimes April. Schedule varies a little bit. And when we release new versions of the site, we'll have both new, enhanced, improved versions of a given existing database, and we'll have new genomes as well. So the number of genomes in BioPSYC has been expanding steadily. And if you would like us to add new genomes, send us a message to the BioPSYC support address. Notice at the bottom of every page, it says report errors or provide feedback. So we're always happy to hear from you. If you do see an error in BioPSYC, we'd be very happy if you reported it and included a literature citation to tell us a source for the information you're reporting. And if we're able to, we have a minute or two for questions, so I'd be happy to take questions. Peter, if we're able to turn on audio for participants, or else you may have to put them in the chat window.
I have a number of questions. Yeah, I do have a number of questions in the chat. I've answered most of them, but one of the questions was just, where is the list of subscribed institutions? I couldn't remember where to find that. Maybe we can send that later.
Yeah, I'm not sure. So an easy way to find out if your institution already subscribes, there are more than 100 institutions that do subscribe. An easy way to find out is to make use of a subscription. You want to be connected to your university's VPN, virtual private network. So the way we recognize subscribers is according to your IP address, and so your internet address. And so if you're connected to the VPN, that usually will tell us, BioPSYC will recognize that you're a subscriber and give you unlimited access. And typically, you'll see in the upper right corner, see how, right here, see how it says SRI International Subscriber. It's recognized me as coming from SRI. And if your institution is subscribed, it will put something similar up here. If you don't see the name of your university here, there are two explanations. One is that it doesn't subscribe. The other is sometimes our database of IP numbers is incomplete. So you can still check in to see if your university does subscribe. Yeah, other questions?
So another question we had from several people is, how do curators deal with conflicting publications?
Yeah, what they'll generally do is, in some cases, it'll be fairly clear that, say, one publication supersedes another, that the person who wrote the later publication was aware of the earlier results and did an experiment that more or less proved that the earlier results were incorrect. And in that case, we'll often describe the later results and treat that as the correct information. In other cases where there is really a conflict and we just don't know, haven't resolved that conflict yet, the curators will note that there is a conflict and cite both sources of information. Other questions?
Let's see. Yeah, I guess there is also another person. So if my institution has the subscription, do I also need a license? Can you please explain the difference between a subscription and a license?
Right. So the subscription gives you unlimited access to the BioPSYC website. If you want to access the downloadable file versions of BioPSYC, then you need a license from SRI. And the way to get that is if we go to biopsych.org, and at the very bottom, let's see, if we click on the bottom on software data downloads, you use this page to request a license from SRI to obtain the BioPSYC files. Now, I'll also mention that my group also produces a software system called Pathway Tools that is the software behind BioPSYC. And if you install Pathway Tools locally, which does require a license, but Pathway Tools is free to academics. If you install Pathway Tools locally, you can process your own genomes with it to create BioPSYC-like databases. So if you've sequenced a new genome in your lab, you can run it through Pathway Tools to create a PGDB, a BioPSYC-like database with predicted metabolic pathways, predicted operons, et cetera. You'll be able to combine it with PGDBs for other organisms, run comparative operations, and use it to help analyze the genome.
Okay, I think that's all that we have. There's one person who was interested in how to train to be a Biosight Curator, but I don't know if you have anything to say about that.
Well, we do have a curator guide. If you Google BioCyc Curation Guide, I think you'll find our online curation guide. And we actually are developing web-based tools for curation that would allow remote groups to contribute to BioPSYC. So if that interests you, please contact one of us through email.
Okay, actually, looks like we have a list of the subscriptions. I'll go ahead and paste that list in. Actually, yeah, it's actually, yeah, it may be okay. This is on the Q&A, so I'll send out the list of subscribed institutions. I think that's everything we have.
Excellent. Well, thanks everyone, and hope you can join us a week from today for more information about some of the more advanced capabilities of BioPSYC.